tomography and its application. In this occasion, we are honored to welcome our speaker, Dr. Nie Xin from Thermo Scientific, Dr. Alexander Ludwig from Nanyang Technological University, Dr. Yudhi Nugraha, researcher and the Esman Molecular Biology, Research Center BRIN. Our moderator for today, Ibu Fina Amreta Laksmi, STP MSc PhD, researcher from Research Center for Applied Microbiology BRIN, and all participants who have attended this webinar. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera, and for the committee to take the screenshot. Be ready. I will start count from first slide. Okay. 1, 2, 3, and the second slide. 1, 2, 3, and the third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat feature. Ladies and gentlemen, now we are going to start our discussion for today, which will be led by Ibu Fina Amreta Laksmi. To Ibu Fina, time is yours.
Okay, thank you very much, Pak Tatang. So today we will start our session with our honorable speaker. So today we have 3 distinguished speakers. So we have like a total of 3 sessions. In the first session, we have Dr. Nie Xin from Thermo Scientific, and then in the third season we have Dr. Alexander Ludwig from Nanyang Technology University, NTU, and in the third session we have Dr. Yudhi Nugraha from the Research Center of Molecular Biology Eijkman in BRIN. And then we also have a Q&A session, discussion session with our distinguished speaker here. So if any colleague who join today's webinar want to discuss with our distinguished speaker, we are very welcome to join you all in the discussion session. So without further ado, I would like to welcome Dr. Nie Xin in the first session. He will present the presentation with title Exploring the Hidden World of Cellular Architecture with Cryo-Electron Tomography. But before I welcome Dr. Nie Xin to the stage, I would like to share his curriculum vitae. So for the colleague you here can know him more before his presentations. Give me a moment. So hopefully you can see the CV that I share with all of you in here. So here is the biography of Dr. Nie Xin. So Dr. Nie Xin is a life science product specialist at Thermo Scientifics, specializing in cryo-electron microscopy, cryo-EM workflow application, including single particle analysis, cryotomography, and microelectron diffraction. With extensive experience in cryo-EM, Dr. Nie Xin has successfully assisted various customers across Asian Pacific continents. Based in Singapore, Dr. Nie Xin is responsible for driving Thermo Fisher Life Sciences electron microscopy business development and growth in APAC for academia and pharma. So I believe that we can gain a lot of knowledge here from Dr. Nie Xin. So without further ado, Dr. Nie Xin, I welcome you.
Thank you, Dr. Fina. Thank you very much for your kind introduction. So now allow me to share with you my screen and we can start the discussion from here. Let me share this one. Okay, morning everyone. And Dr. Fina, are you able to see the screen I share over here?
We can see your screen.
All right, great.
I think
Thank you.
still in the presentation in your side mode, I guess, because we can see two.
You can see two screens. Okay, let me change that. Okay, is it getting better?
No, it's perfect.
Okay, great. Thank you so much. Good morning, everyone. So thank you, Dr. Fina, and also the whole team for BRIN inviting me to join this webinar. So today I'd like to share with you a general overview about this relatively new technology called cryo-electron tomography, and how can we utilize this workflow to understand particularly cellular context with particular high resolution of the structural information hidden within the cells. So before we start, I'd like to share with you, we're coming from the Thermo Fisher, which is one of the leaders in the life science industries. And the mission of the company is to make the world a healthier, cleaner, and safer place. And for people who has experience before, you understand that last year we have already hosted one big workshops with Cryo-Em, with focusing on single particle analysis. On single particles, we can understand the structure of the proteins. But in this case, the sample we are understanding can be isolated proteins, purified, or maybe purified virus particles. But actually, in life science, it's more complex. A lot of proteins need to interacting with each other at the cellular context. So today, I'd like to share with you another workflow we call the cryo-electron tomographies, which can help us understand the protein functions within the cellular context. But of course, maybe in the future, we can discuss further with also the electron diffractions, which also understand the structure of the protein crystals. So together with electron tomographies, we can able to see the whole pictures in situ, which means we are not only able to see individual proteins or maybe individual protein complex, we are also able to understand how this protein complex interacting each other at the cellular context, as a native environment. For example, there's a couple of examples over here. The first example is actually from the coronavirus. We are taking the cryotomographies of the live coronavirus, which is a BSL-3 levels. So at this stage, you are able to visualize the spike proteins on the surface of the virus particles, and especially how the distribution of the spike proteins on the surface, and whether we're also able to understand at a much higher resolution of the structure information to see whether they're presented in the pre-fusion or post-fusion isoforms, and what's the angles of these all the projections relatively to the surface of the virus particles. So a lot of information can be understood from one tomograms. And also, if you are able to see what happens at the cellular context, you can see that we can open up the cell and able to observe that how the different organelles of different protein complex are organized and interacting each other within the cellular context. You can see the protein complex, the ribosomes, and also some ER binding, some free-floating ribosomes within the cells. And there's a lot of advanced technological improvement in the recent years. So if you go back to history, almost maybe 10 or 20 years ago, we are only able to see very rough understanding of with tomographies. But with the new technologies, new hardwares, new softwares, new sample handling methodologies developed, we are able to improve the resolution. We are able to see much higher. So now we are able to really understand the protein complex in situ together with our new technology called cryo-focused ion beam, also cryo-clamp, which is a correlative between electron signal together with the fluorescence signals. And also there is a lot of improvement on the hardware side and softwares, including different software for data collections and for data processing. So over here, I listed out, actually this paper listed out the recent breakthroughs or maybe new advance in this cryo-electron tomographies, including both the software side and also the hardware side. From hardware, you can see we have this developed the cryo leaf-out and the waveform method for some of the sick tissues or maybe some of very large volume samples. And also we have integrated this integrated fluorescence light microscope inside our cryo-feed chambers, which can help you correlating the fluorescence signal together with SEM signals. And also in the software side, we have this newly developed Tomo file, which is a really fast and convenient data collection softwares. So you can see, especially in the recent years, from 2020s all the way down, more and more technologies are focusing on the cryo-ET or cryo-electron tomographies, especially the new advanced sample preparations called cryo-feed laminar preparations, and also the Tomo file for data collections. So with this, you can see we have a wide range of applications can be understanding from these workflows, including in the most likely to be the cell biologists, then also in the microbiologies, biologies to understand how the virus replicates itself within the host cells. In neuroscience, it's also important to understand the synapses, how the neural cells transmitting signals from one to each others. And also in plant biologies, we have a lot of applications to understand the photosynthesis or the plant immunities, a lot of important for the agricultural research, and especially for the biomedical and the drug discoveries, using the structure informations to understand the structure-based drug design and how the drugs affect the cellular response to different drug targets. So you can see we have a wide range of applications. So I hope from this seminars, scientists from the audience are able to correlate your own research together with Cryo-EM. I hope Cryo-EM can help you research taking one step further. So also this cryo-electron tomography has been listed as a method to watch in the Nature method in last years. Because utilize these workflows, we are able to understand and also visualize the proteosomes within the cellular context. You can see that previously, if you want to see the proteins, you need to purify them from the cellular context, either from the single particles or maybe from mass spectrometry, NMRs, you understand. But the protein is much more complex or complicated than we imagined. So if you look at the protein complex as a cellular complex, we call the visual proteosomes. You can see the nuclear pore complex, or the cellular matrix, how the ribosomes are attached to different vesicles, or maybe attached to ER membranes, how the nucleosomes was organized and forming the nuclear pore nucleosomes, maybe the we call the different nanometer fibers, how the histones were organized at the nucleosome complex, and also how the mitochondria responds to different stress. So all these difficulty informations we now can able to visualize at a cellular context. We call it the visual polynomials. And also from this, we are able to resolve the novel structures, which maybe people do not understand or do not able to see from the previous technologies. And also we can understand our organelles in a much higher resolutions. Previously, maybe we do a lot of stainings with confocals. You see different fluorescent signals. But now we are not limited to see only the signals, but we are able to see the structures, how the protein was located on the surface of membranes, how the vesicles, what was the content within the vesicles. And also we can visualize the protein distributions, the dynamics, how the protein is interacting with each other at a cellular context. So you can see that we take one step further to understand life at the 3D together with electron tomographies. So over here, I like to share with you, this is a very simple drawing to show you the basic workflows, which means how the sample was prepared and collected with this workflow. So basically, we are separated this into two different scenarios. So the first scenario is with relatively thin samples. So I hope the audience can keep in mind that the electrons is the limitation to penetrate your samples. So only a couple of hundreds nanometers is the maximum limitations. So for some of the smaller samples, including like maybe protein complex or reconstituted protein complex together with a viral, together with a lipid vesicles, or maybe some of the virus particles, purified organelles, or maybe relatively thinner parts of the cells. So in this case, all the samples are relatively thinner enough. We are able to visualize all these samples directly. So which means we can, very close to the single particle workflows, you can vitrify your samples with a vitrobot, which is a robotic machines to help you do the vitrifications, plant freezing into the liquid eatings, vitrifications. They can collect your tooth series using the Cryo-TEM. So in this case, two major tools are needed. The first is a sample vitrification, including the vitrobot, and second is a Cryo-TEM. So in BRIN, they have the latest model of the Cryo-G4, which is the highest setups of the Cryo-TEM. So you can collect the whole tomograms within the TEM and reconstruct it in 3D using the software, so reconstruction, segmentations, and getting 3D understanding of your proteins complex, or maybe some of the cellular context. But majority of samples, actually, to be frank with you, is organized in 3D. Even the culture cells, which is too thick for the electrons to penetrate. So in this case, we need additional tools because of focus ion beam, which can help us to thin the cell, which is make it thinner enough for the electron to penetrate. So over here, you can see we have this one particular example is from the some of the green algae is the membrane PBS PS2 complex. It's a really regular organized protein complex for photosynthesis. But of course, the culture cell is really thick. It's difficult for electron penetrate. So what we need to do is first we plant freezing the samples on the surface of the TEM grid, utilizing the focus ion beam. Also, it's possible maybe we can do some correlation with the fluorescent information to figure out which is our region of interest. I utilize the focus ion beam to make our samples thinner enough because the lamella are cryo-fin millings to produce really a thin slice of the sample. We call the cryo-lamella. So this lamella is in the thickness in maybe 200 nanometers, which means the electron can penetrate through these cryo-lamellas for the electron tomography data collections. So after collecting the tomographies, you can see that we can understand the whole protein structures of this PBS PS2 protein complex in a much higher resolutions to understand within the cellular context how the protein functions for the photosynthesis. So in these workflows, you need additional tools we call the Aquilos, which is our dedicated cryo-feed platforms. Actually, we have multiple platforms in the cryo-feed field, including Aquilos 2, which is equipped at BRIN, and also with this latest Hydra 5. This is a plasma feed platform, and also the Actis is also our fully automated cryo-feed platforms. But in these workshops, the participants are able to experience the operation from the sample verifications and also the operation of the Aquarius 2, which is our dedicated cryo-feed platforms, and also data collections on the Aquarius G4, which is the cryo-TEM. So you can see this is the illustration of the workflows which I shared with you just now. So your cultured cell, maybe your purified suspended cell cultures, will be plant freezing with a vitribot, which is step we call the vitrifications. And as this sample will be transferred to the Aquarius for the lamella makings, and also after the lamella was ready, which is a thin, really thin layer of cellular tissues suspended, which is electron transparent, will be transferred to the Cryo G4, Cryo-TEM for the tomography data collections. So this is really quite straightforward workflows by including 3 different tools. So from this, I like to play a short video to give you an understanding what is the operation looks like. The first example is plant freezing on the surface of the TEM grid. And this TEM grid will be transferred into the chamber of the Aquarius through a cryo shuttle. So over here, you can see that this cryo shuttle was transferred into the chamber of Aquarius. And Aquarius we call the small dual beam because it's including the electron beam as a SEM and also the focused ion beam. So the first step of this operation is to utilize the GIS as a protective layer on the surface of the samples. So after the GIS coating, you can see that your shuttle is over here, will be imaged by the electron beam. And the focused ion beam will help you remove the materials from your samples and make it thinner enough for the electron to penetrate. So this is one example of the frozen hydrated cell on the surface of the TEM grid and utilizes the focused ion beam. We can remove the material. You can see there's 2 different windows. One is on the top, the second one is on the bottom. So all the materials between these 2 windows will be removed. So you can see only a thin layer suspended in the middle. So this part we call the cryo lamella will be preserved for data collections. So after cryo-feed millings, this lamella is ready. So we need one step further, we call the protective platinum coating, or we call the spot coatings as one layers to protect the surface of the samples and also make it more conductive. But all this operation you can see is will be finished within the chamber of your Aquarius. So actually there's no need to take your sample in and out, transfer, and eliminate any possible contaminations. So after the smarter coatings, you can see the surface was covered with one layer of the platinum as a protective layers and also make your sample more conductive under the TEM. So afterwards, the sample will be transferred to the Cryo-TEM. And also you can see the sample was in the same format. We call the auto grid. You can connect it easily from Aquarius to the Cryo's TEM. So inside the TEM, you can utilize the tomography softwares for the automated data collections. So you can see we are collecting your samples from different angles. We call the tilt series. So from the tilt series, we can reconstruct it a 3D volume. So you can see this is the illustrated, reconstructed, segmented beautiful 3D volume. So from one slice of the samples, you can get in a 3D volume, help you understanding the cellular context at the 3D. So from here, I like to give you some highlights of each of the steps in these basic workflows. So the one more important steps that is focused ion beam, we call the Aquarius, which is I show you in the beginning of the cartoons. So this Aquarius will be including 2 important parts. One is this SEM for the imaging purpose, and the focused ion beam, FIB, is for the milling purpose. So milling, as this cartoon shown, is to remove the material of the samples. So which means you can open up the particular area of your cells, and you can do the imaging of your regional interest, which is seen. This is a cryo lamella suspended in the middle. And this Aquarius is actually widely adapted worldwide. I think currently there's more than 100 systems installed all over the world. And you can see more than a quarter, which is 26% of the publication which involve Aquarius are all going to the top journals, including the Cell, Nature Science, the CNS top journals. And in these platforms, you can do multiple jobs. So what I show you just now on the cartoons is mainly for the lamella preparations, which is actually fully automated. laminar preparations. And also we have the integrated fluorescent microscope, which means you can check both the fluorescent information together with your ICM imaging, do the correlation, and also help you figure out your region of interest, and also the expression levels, also to see where the fluorescent signal come from. And in addition, which will not be covered in these workshops, but maybe in the futures, is we can reaching out really huge volume. We call the cryo-leaf-out, which including cryo needles and the leaf-out workflows, and also the slice and views. So the most important functionalities for the Aquilus, I would say, is laminar mean preparations. The cartoon looks a bit complex because including a lot of operations, looks M beam, S beam, and also the coatings, a lot of steps. But actually, the whole steps was fully automated. What the customer need to do is just to tell the software, or tell the system, where is my cell and where is my region of interest. After selecting the region of interest of your cells, the rest job will be done automatically by the software. You can see this is a nice example. Is this customer selected roughly maybe 20 different sites. Okay, this is how the TEM grid looks like, and this cell growing on the surface, prepared on the surface of the TEM grid. So customer only need to tell the software, okay, this is the site I want to do. This is my samples. Then the rest job will be done. All the milling process will be done automatically by the software. So you set up the workflows, you come back next mornings, about 20 laminars are ready for the data collections. So in addition, you can see that we are able to visualize the fluorescence information from the Aquilus. So this is including integrated light microscope inside the chamber of the Aquilus. So you can combine both ICM signals together with the fluorescent signals. So this gives you a lot of flexibilities and advantage because you can easily locate region of interest. And also there is no need to moving in and out for your samples. So eliminate any possible contamination risk. And also you always can check in the middle and afterwards your film milling process to make sure that your fluorescent signal was preserved as a laminar you prepared. Make sure what you're imaging on the tomogram is exactly what you're looking for. So now you can see just now what I shared with you is some basic workflow on the laminar preparations. So in addition, maybe in the future, we are also able to perform the cryo-leaf-out, which is handling some large volume samples, including the cells growing on the surface grid, or even some tissue samples, like maybe the brain tissues, kidney tissues, or even the whole volume of the organism, like maybe C. elegans or Drosophila egg. All these large volumes can able to access from the cryo-leaf-out. So after your sample is ready, the next step is to imaging under the TEM. So this for the data collection. So this including both the hardwares and softwares. So hardware-wise in BRIN, they have this latest high-end Cryo-TEM, Cryo-G4. And the software-wise, we have the Tomo 5, which is a really easy data collection platform, and also the Tomo Lab for the after data collection for the data reconstructions. So the first of all, the Cryo-G4 is the ultimate with the best imaging quality platforms so far. So including both the selectress and the filter and the Falcon 4i imaging cameras. So combining these two, all these three important parts together, they give you the best image qualities and highest resolutions, especially impactful for cryotomographies. So you can see this is one example taken tomogram from the selectress and the filters. You can see very clearly all the vesicles related with these Golgi apertures. And you can see that on the surface of vesicles, all the different covalent protein complex, you can see how the enrichment of covalent protein complex on the surface of vesicles directly without any of the illustrations. You can see directly densities on the surface of the covalent complex. And over here, you're also able to see some of the budding vesicles over here on the surface of the protein complex. So for the data collections, we have this Tomo 5 softwares. So I'm not really into each of the steps now for data collections, but I want to highlight over here is a really simple method. So for the users, there's no need to learn the really difficult operations of the softwares because it's almost fully automated workflows. So one important highlight I give you is over here is first, the Cryo is equipped with autoloader, which means you are able to logging up to 12 different samples in one batch, which means it's not one sample, two samples, but 12 different samples you can put into the microscopes. And the Tomo file has the capability to screen all the 12 different samples one by one automatically. So user no need to vary which is how to handling to the samples to check the sample qualities. The software will help you do the sample quality checking automatically. Just one click, they do the grid mapping for all the 12 different samples. So after set up your recognize different lambda sets, you can do the automatic batch tomos, which means the users also no need to sit next to the microscope. You just select, okay, 1, 2, 3. These are 3 different lambdas. You set up different locations on each of the lambdas, and the rest job will be done by the software. So multiple tomogram will be collected automatically. We also recently has integrated, this is called the multi-shot capabilities, which means even within the single cryo-lambda, which is prepared by the Acros, we can collect multiple tomograms within a one session setups. You can see we only need to set up one like focus areas. Then you can have a multiple area which in green for data collections. So this is really expanding both the speed and the available capability for the imaging areas within the single lambdas. So give you a really huge advantages. There's no waste of your imaging areas, but try to preserve as much of data as possible from single lambdas. So after the data collection, we are going to acquire one or multiple tilt series, which is shown over here. However, the tilt series only give you the raw data, which is how the image looks like from different angles. So we need additional steps to do the reconstructions of from this tilt series into a 3D volume, we call it tomogram. So from tilt series to tomogram, this step called the reconstructions. They used to be a really complex setups, but now we have this new Tomo Lab softwares. So within the Tomo Lab software, there's user no need to worry how to set up the reconstructions because it will be done automatically on the fly. So you can see after the tomogram tilt series was collecting finished, the Tomo Lab will reconstruct the whole tomogram reconstruction automatically for you. So you can see on these illustrations, over here, the first image is a raw tilt series. So you can see a little bit jiggling because of the tilting at each sample maybe shifted a little bit from each of the steps. But all the different steps images will be aligned automatically within the tilt series. And also the Tomo Lab will do the reconstruction volume for you automatically, which means while your tomogram tilt series will do the collections, when the collection is finished, just 5 minutes, your whole reconstruction volume is ready for you to view. So the volume is ready. So there's no additional operation needed, no third-party software needed, just to check your result immediately. You can see combining the Tomo Lab software together with the Tomo file data collections, you can easily get a high throughput rate, very easy data collections, and you always preserve the best images from your tilt series. So after the tomogram was collected, finished, we get this grayscale 3D volume. You can see if you have some of the experience, you can directly visualize some of the key features from these tomographs, including the nuclear complex, the Golgis from all the ER membranes, and also the cytoplasm. You can see some vesicles, even the ribosomes from cytoplasms. But however, this is only in grayscales, but we need to highlight whatever we are interested. So we have additional step we call the segmentations, which will be done by another software called Amiras. So with Amiras softwares, we can segment all these grayscale 3D volume into a more colorful, more representative 3D volume. You can highlight your cellular features, all the different membranes, the cis Golgis, the trans Golgis, the nuclear complex, all the different vesicles in different colors. So this is much easier for the scientists to present and interpret their scientific result in the publications and also in the presentation to other scientists. So this is one important step called the visualization and the segmentation of your samples. So this is all the basic steps that I share with you from sample prep, data collections, data processing, and also segmentations. So in the couple of minutes, I just give you some of the good examples that we're doing for data collection using this tomogram workflows. The first important example is actually this is the coronavirus replication itself is within the whole cells. So after the virus infections from the tomogram, the scientist is able to visualize this double membrane vesicles, which is very important for the viral genome replications. And also the visualize on this double membrane vesicles, there is a dedicated structure, is a chrome-like structures, which is important for the release of the viral genome after replications. So scientists has getting from the tomogram, they are able to visualize this particular pore-like structures for some origins, getting the structures, and the hypothesis that this is a very important structure for release of viral genome after replications. From this, we can design particular small molecules or some of the particular drugs to targeting these structures. We call a structure-based drug design to very specifically to inhibit release of viral genomes, thus inhibit the infections of the virus to other cells. Some other examples is also to visualize a protein complex as the surface of virus, including the HIV envelope glycan proteins. You can see the difference between the immature virus particle and also the mature virus particle. You can see the different localization and structural information of the glycoprotein on the surface of virus particle. But in the more cellular context, we can do the field meetings on the multiple cell connectivities to see how the cell-cell connections will get it. You can see this is a sample prepared between two different cells, and you can get the tomograms exactly at the location of the cell junctions. So from this, you can see all these molecules was located at cellular junctions to transfer materials and transmit signals between cell to cell, and it's very important for cell-cell signaling. So one additional information is we are able to obtain high-resolution structures. We call the visual proteosomes, which I shared with you just now. You can see this is one example is ribosomes, which is important target for a lot of drug antibodies. We can get in the high-resolution structural information of the ribosomes within the cellular context. So this is an example taken from yeast cells. You can get in the subatomic origin of the cellular context, getting the structural information of the ribosomes and how the structural information looks like within the cellular context. So currently we are initiating, this is one huge project, which is called the Visual Proteasome Project. This is including a large number of tomograms, more than 2,000 tomograms. And data collections was on the Quails G4, Selectrix energy filter, and Focus 4i cameras. So from this, we targeting to imaging all the proteins within this model organisms called the chlamydomas. You can see this is one dedicated example. You can see all the different organelles. But organelles is only the cellular context structures. We also want to understand which proteins are located within the cellular context. You can see so far we're getting multiple protein structure of the result, including the ATP synthesis, different ribosomes, rubiscos, and also nucleosomes as a cellular context. But this is really a huge project, including a lot of laminar preparations and tomogram data collections. So over here, just highlight with you, this is a new machine that we're doing this project called Actis. So this Actis is also a very powerful plasma feed-based cryo-feed milling platforms. It is equipped with autoloaders, which means you can handling up to 12 different samples within one loading of those samples. So ensure the high throughput. And also the autoloader system was exactly the same as the Quails, which is ensure the connectivities. And also they have integrated fluorescence microscope and high power plasma feed for the batch sample preparations in these Actis platforms. So I think this is what I'd like to share with you today is a general overview of the Cryo-Electron tomographies, how the sample was prepared, data collected, and processed. And I hope to give you some basic understanding of the tomograms, and this can be maybe make some synergies of your current research project. So for these workshops, we are very welcome all the audience participants to submit your samples to BRIN and for the workshops. But of course, there's a basic requirement for the tomogram data collection for samples. So you can see over here, this is an example of different samples as available for the Tomo data collections. Something really small, the virus or large protein complex, purified organelles, bacterial samples, fungus, yeast, or maybe culture cells. And something really huge, even the organoid tissue samples, or even some of small organisms. But for instance, in these workshops, there's a certain criteria for the samples. So there is a list like the culture cell, the thickness should be less than 10 micrometers, which means they are if it's too big, too large, it spend a lot of time for the field meetings. So for the efficiency for the workshops, we require the samples to be less than 10 micrometers thick. But I believe most of the culture cell will be less than 10 micrometers in the thickness. And of course, the suspended cell culture is preferred because it's easily prepared on the sample vitrification process. But you're also capable for prepare some of the culture cells. But this including additional steps because you need to culture your cell together with the TM grid. But I also welcome you to submit your proposals. So our application specialist can help you for the sample culturing and the sample preparations on different steps. And however, for some of the sick samples, which is tissue samples or maybe some organoids, it requires a high pressure freezing. But in this case, we are not including high pressure freezing in these workshops. So you should not submit too big samples in these workshops. So in a quick summary, so this is our preferred samples in this workshop, including some fungal samples, suspended cell cultures, yeast samples, or maybe the culture cells on the surface of TM grid are capable for these workshops. So in this, we can prepare the laminas on the cell cultures and open up cells and do the tomography data collections, data processing on your particular samples. Okay, audience, I think that's all I would like to share with you today. Thank you very much. Please keep your questions in the Q&A sessions. Okay, thank you, Dr. Fina. I will pass it back to you.
Okay, thank you very much, Dr. Nie Xin, for your insightful presentations. So before we can open any discussion between our colleagues with you, Dr. Nie Xin, we can proceed to other distinguished speaker first. So we will finish the presentation session first, and then we can have discussions.
Yes.
Thank you very much, Dr. Nie Xin. Okay, so after Dr. Nie Xin, we still have other two distinguished speakers. So the second distinguished speaker is Dr. Alexander Ludwig from the Nanyang Technology University, NTU. Maybe I would like to read some of his work. So Dr. Alexander Ludwig is from School of Biological Sciences and NTU Institute of Structural Biology, Nanyang Technology University in Singapore. So today, he will present his presentation with title Native Architecture of the Human Inactive X Chromosome Studied by Correlative Light and Cryo-Electron Tomography. So my dear colleagues, I hope you can gain inspiration from the research that will be presented by Dr. Alexander Ludwig. So I would like to welcome Dr. Alexander Ludwig to the stage. Time is yours.
Thank you very much. Okay. Somehow I have a problem with sharing my screen.
So, or you can share the presentation with us and we can share for you.
Somehow I'm I don't see. Do you see my screen?
Yes, we can see clearly.
Is it in presenter mode?
No, now yes. Now
Oh,
or
now.
in the
Wonderful.
presentation.
Okay. Technical issues as always. Yeah, thank you very much for the invitation to share some of our recent unpublished work. So I should start by saying that I'm a cell biologist. I'm not a structural biologist, although I'm working in a structural biology community at NTU in Singapore. So we're part of this Institute of Structural Biology. Many of my colleagues use cryo-EM, single particle cryo-EM mostly, to study their most favorite molecules. We come into the picture from a cell biology point of view. So we're interested in cell architecture, and we've been using different EM techniques for many years to address the problems we're interested in. So what I thought I'll do today is to share with you some of the experiments we've done on looking at the native architecture of a specific chromatin domain inside cell nuclei, mammalian cells, cell nuclei, using correlative light and cryo-electron tomography. So, of course, a few words about the background of the story, the biology. I hope you're all familiar with the concept of how chromatin is organized in the nucleus. So we have of course the principal building unit, building block of chromatin, which is the nucleosome or the nucleosome core particle. This particle has been solved by single particle cryo-EM and X-ray crystallography. It's a spherical particle, round or disc-shaped, and we have very detailed information about how this nucleosome core particle is organized, how DNA is wrapped around this histone core. Now, the next level of compaction would then involve mechanisms that are not quite so clear. So somehow these nucleosome core particles are arranged in strings, of course, and there is the speeds on a string model that's mostly derived from looking at isolated chromatin by negative stain EM, for example. And this then would sort of, in a way that's really not understood, be organized in chromosome territories that we find in situ in mammalian cell nuclei, where each chromosome is believed to occupy a certain area in the nucleus. And some of these chromatin domains are, of course, active, so they're transcriptionally accessible, and others are not and are silenced. So there's interest, of course, has been for many years, decades, in understanding how chromatin is really organized inside the cell nucleus. So this is what we're trying to address in part in this project that I'm sharing with you. So what I should also say is that although cryo is very powerful, as Nie Xin has just shared with you, and I'm going to share with you later, it doesn't always have to be cryo-imaging. So you can visualize chromatin using other techniques, conventional TEM, for example. So in one of our latest articles, we used a staining method called APEX. So APEX is a peroxidase that you can fuse to your protein of interest. And in this very simple chemical reaction with diaminobenzidine, you can create electron density around your protein of interest and therefore visualize where your protein actually resides inside a cell or tissue section. So we use this approach to study telomeres, which are the end of chromosomes in mammalian cells, is another very specific chromatin domain in mammalian cells. So we were interested in visualizing this domain using this APEX labeling technique and electron tomography. And all of this was not done under cryo conditions, but in plastic embedded, stained, and dehydrated samples. So for those of you who are interested in this in more detail, please, I refer you to the article, but just a few highlights, this one slide to show that we can visualize these telomeres in cell sections. A typical 2D image of a relatively thick section would look like this. So you see the telomeres labeled here through the electron-dense stain that we introduced with the apex probe. And after tomography, you can see, of course, you can determine much more detail. You see actually individual chromatin fibers inside the telomere. So that allowed us to overall describe the architecture of telomeres and start to measure how thick these fibers actually are, these chromatin fibers that we find in telomeres, and how they are arranged inside the structure. So with this in mind, we, of course, were interested in other chromatin domains. And at the time, we also then had the possibility to do this using cryo approaches. So another chromatin domain that we were already studying before we started using the cryo approach is this bar body, or the inactive X chromosome. So the inactive X chromosome is, of course, only present in female cells, and it's a chromatin domain that is facultatively silenced. So it's a facultative heterochromatin domain. It's important for gene dosage compensation in mammalian cells. And you can clearly see that this inactive X chromosome here looks quite different from the active X chromosome in female cells. But the exact mechanism as to how this one chromatin domain is silenced is not really understood. So, of course, people have been interested in this for a while, but we don't really know much about the architecture of this domain. People have been using conventional TEM to look at this bar body. Here, for example, is a series of 2D images of the bar body stained with conventional heavy metal stains in plastic embedded sections. But really, how chromatin is organized in this particular region in the nucleus wasn't quite so clear. So our goal was to study the architecture or the ultrastructure of the bar body using Cryo-Clem. But before we started Cryo-Clem, we were actually again using this apex probe that I just introduced and conventional TEM. So we generated a cell line. These are retinal pigment epithelia cells. These are female cells. So they have 2 X chromosomes, one of which is silenced. And we expressed in these RPE cells an apex probe of the histone variant macro H2A. So this histone variant is specifically targeted to the inactive X chromosome, as you can see here. So this protein then had a GFP tag that we can use to visualize the protein by light microscopy. And it had this apex tag that we can use to enhance the contrast of the inactive X chromosome in TEM. So we used a correlative imaging approach for this, which sort of looks like this. We start off with the GFP signal of fixed cells, here indicating the bar body. Then we would perform the apex reaction, plastic embedding, sectioning, producing relatively thick sections, and would monitor where the bar body is in these sections. So here is a correlative TEM image of this area. Then we would home into these areas and perform conventional room temperature temp. So we did a lot of 2D imaging, but we also did tomography on this bar body. So just as a reminder, Nie Xin has already introduced tomography. What you do in tomography is that you tilt your sample plus minus 65 degrees. At each incremental tilt angle, you take a 2-dimensional projection image of your sample. And once you have this tilt series, you can reconstruct this into a tomogram to obtain a 3-dimensional representation of your object that you've been imaging. And of course, this is very powerful for any kind of things that are happening in a cellular context. So to study rare objects, irregular shaped objects, or things that you cannot easily isolate. So this is then a typical tilt series of a bar body here stained with this apex probe. And on the left and on the right is the reconstructed tomogram. of the center region of the bar body, and you can see that these chromatin fibers here stand out, so they are more heavily labeled than the surrounding, and so we can start tracing these chromatin fibers in 3 dimensions, which is actually quite a challenge. So just to show you what you can see with these in these tomograms in this stained approach, you can, again, actually appreciate chromatin fibers, and you can start measuring how thick these fibers are with, of course, the resolution limit that you have in conventional stained TEM samples. But I hope you can appreciate that we can clearly trace these fibers. They are somewhere between 20 and 30 nanometers thick. Of course, we don't know how many fibers this actually represents because of the resolution limit, but it was a good starting point for us to understand how big this bar body actually is, what it looks like inside a nucleus, and so provide a good foundation for the cryo work that I'm going to share with you now. So there is a problem, especially in cryo, and that is to find your target. So as you might know, in conventional TEM, you can produce serial sections of your cell or tissue. And so if you think about just one cell, which is maybe 10 to 20 microns across in volume, you can produce single sections through your cell and then image basically each individual section. And without prior knowledge where exactly in the set volume your object is, you will find it eventually. Plus, in the samples that I just shared with you, they were stained. So you can actually see where the bar body is simply by the increase in electron density in the nucleus. In cryo, you don't have stains, or at least no well-established electron-dense stains that would guide you directly to your target. So finding the needle in the haystack, as people often say, is really a little challenging. And in our particular case, of course, we were interested in a specific region in the nucleus. And so how do we find our bar body back in vitrified samples without actually having an electron-dense stain in our tissue sections? So that is, of course, a technical problem, and Nie Xin already shared with you how we would go about fixing this problem. And this is by fluorescence-based or guided cryofit milling. So in our particular case, we would have these RPE cells expressing macro H2A, so the bar body marker in our cells. So this would produce a fluorescent dot in the nucleus. And the idea was to image these cells, these vitrified cells, on a specialized microscope, light microscope, the core side, to find the target cells and the bar body within the target cells. Having done that, we would transfer our sample to the Aquilos CryoFit SEM. We would find our XY coordinates back, and we would produce these lamella that Nie Xin talked about. So expose our bar body in this lamella that we can then image by cryotomography, which in our case was a Titan Cryos. So, just to mention, this workflow is kind of more complicated than the one that you can actually you have available at BRIN, because at BRIN you have an IFLM, so a fluorescence microscope in the Aquilos column. So that makes your life actually a little easier in the sense that you don't have to worry about this particular instrument anymore. You can simply load your sample straight into the Aquilos, find your target area, as long as you have labeled it with, for example, a bright fluorescent protein, and that would directly guide you to your place that you need to expose by cryofit milling. We don't have this IFLM addition on our Aquilus. We have instead this light microscope, which of course in our case is an additional step that we need to conduct. So nonetheless, this actually works quite well. So this is what we see on the cryo stage on the core side instrument. So what you do here is on top of here, so this is an inverted light microscope. You come from the bottom with a 40x air lens with an MA of 0.9. And on top of here on the stage is typically for cryo-imaging, a cryo stage that you can cool down with nitrogen gas, such that your frozen, your vitrified samples, your grids, are maintained vitrified. So you're imaging here under cryogenic conditions. So you can obtain grid overviews by transmitted light, and then you can home into different areas here and visualize your sample with 4 different laser lines. So we can have anything between DAPI and far red. We can image both by wide field and confocal imaging. And this is just an example here of one of our frozen RP1 cells expressing this macro H2A GFP marker. You can clearly see here that the bar body stands out. You see the nucleus. And you can also appreciate here in the red channel, we have fiducial beads that I will talk about in a minute. So having obtained the XY coordinates of our bar body, we can then move the grid onto our Aquilus 2 instrument to perform cryofit milling that Nie Xin has already introduced. The additional challenge here is now, of course, to obtain the lamella right through our target, which is only about 1 micrometer in size, maybe 2. So we would perform this cryofit milling, starting with coarse milling and finer milling to end up with lamella that are somewhere around 150 to 200 nanometers thick and about 6 to 8 micrometers across. So how do we actually obtain the precise X, Y, and to some degree Z coordinates of our target in the cellular volume? So one thing that we have learned is very, very useful are these fluorescent fiducial beads. So they're not only fluorescent, so we don't only see them in the core site by fluorescence microscopy, but they're also electron dense. So they're quite relatively big, about a micron or 2. So you see them as distinct dots by fluorescence microscopy, but you also see them as dots in the SEM. So once you have your target cell and these fluorescent beads, you can overlay the 2 imaging modalities such that once you are on the FIP SEM, on the Aquilus, you have exactly, so this is shown here, this is an SEM image at an angle targeting the area here. This cross is where the bar body is, and here are some of these little dots that are caused surrounding the cell by these fiducial beads. Gives us very much confidence that the target area that we're targeting is really where our structure of interest is. So this is really quite useful, and there's also a 3D toolbox that one can use on the Aquilus to do this. Have been published a while ago. So this is just, again, to illustrate the correlation procedure, and also to say that once you have produced your lamella, you can do a post-correlation to be absolutely certain that the lamella that you've produced, or relatively certain, that this lamella indeed contains the structure of interest. So these are overlays of the finally produced, the final lamella, with the fluorescence overlay of the cell. Here is the bar body. Here's another one. And so when you zoom in, you will see that, yes, the bar body should, for all we know, be contained within the lamella. And B, not only that, is in case should your correlation be precise, you will actually also know where on the lamella you will have to look. And that's quite important for later on. So anyhow, so then we transfer our lamella onto the Titan Cryos. And we set up data acquisition. We did this in a quite conventional way at the time. We had a K2 Summit direct electron detector with an energy filter. Now we have a Falcon 4 with Selectros X. But the data I showed you was collected with the K2 at a certain pixel size, relatively low magnification. We used a dosimetric tilt scheme, and we tried to have an electron, total electron dose per tomogram of no more than 150 electrons per square Angstrom. So we set up data collection on these lamella. This is part of a lamella. We knew the bar body should be somewhere here. As always, at low exposure, low magnification exposure with low electron dose, you don't really see much on such a lamella, especially not when you're operating in the nucleus where there's hardly any contrast. So we set up data collection with and without the faceplate, and the tomograms were post-processed, the movie frames were aligned, and we typically also do denoising using the CryoCare software. All of these things are published by others. So this is a tomogram, finally, of the native bar body. You can see here the nuclear envelope cut in an angle. So the lamella is produced at an angle through the sample. Here is the cytoplasm. You see the ribosomes in here, a bit of the ER. And basically what you see here, all of this dense mesh of chromatin is where our bar body is located. So this is, although recorded with the faceplate at this Mac and zoom, quite noisy. So when you zoom in, you will actually see that we can visualize individual nucleosomes. Here on the left is a comparison to a single particle cryo-EM prep of isolated nucleosome core particles. They are about 10 nanometers in size, and you see disk views and side views. And when we look into our tomogram data, we can actually see these views as well. So this is quite nice. Of course, we're interested now in looking into the more global 3D architecture of these chromatin domains. We find interesting we make interesting observations. For example, we have evidence that these nucleosome particles or units, they often stack with each other into sort of what sometimes even looks like regular arrays. But of course, generally speaking, the overall architecture of chromatin in this bar body is not very regular. So it's very heterogeneous. What we can say, though, is that as expected, that the density of chromatin inside the nucleus, sorry, inside the bar body is much higher than outside the bar body. And we can also, for sure, make the statement that, as always, when you look at native organization of cells, we find structures that we really have no idea what they might be. So, for example, we find larger globular domains that are not nucleosomes, that have sort of a very fuzzy electron density in the core and then more electron dense structures outside, very abundant inside and also outside the bar body. We have no idea what these structures might be. We have some ideas. And so this is just one example as to how rich the data is that you can obtain from such tomograms of vitrified samples. So when you compare our cryotomography data under native conditions, no stains, with our apex stained bar body, you can see the usual thing. You see that the apex labeled plastic embedded heavy metal stained bar body is much more contrasty. You get a better difference between no signal, no density and density. But of course, the cryotomography data is higher resolution. You look at the chromatin and not at the stain, but at the chromatin itself. And so it'll be, of course, going forward, it's challenging for us to understand the architecture and measure things of the bar body, but we're quite excited about the data that we have, because we can learn a lot from it. And actually from both data sets, the stain samples and the cryo samples. So to sum this part up, we have established this correlative workflow in our lab. We can do this on the bar body and on other structures. We have optimized data collection with the faceplate, but I have to say we have had some issues with the Volta faceplate in setting it up and in getting it to work consistently. And we can, of course, now apply this workflow to address other biological problems. When it comes to our bar body data, we are currently collecting more tomography, more tomograms. We want to analyze chromatin organization inside and outside the bar body. We are interested in new developments in the field that can improve visualization and also data tomogram quality, one of which is this missing wedge correction or the ISONET approach that's been published. And of course, we're interested in segmenting and visualizing our tomogram or the things we see in our tomogram using software, Amreta, and other approaches. So just a little glimpse of what else we can, of course, see in our samples. I shared with you the bar body samples, so chromatin inside the nucleus, but of course, we also always have cytoplasm in our lamella and have been collecting tomograms from there. So this is just a snapshot of these two areas from RPE, one cell cytoplasms showing lysosomes, multivesicular bodies, and mitochondria. And with this, I would like to end. I'll skip all this. Maybe we can put these things. I can talk about these things in the discussion. Just thank who actually did the work, which is Barbara here. She's really been the driving force, is a postdoc in my lab, driving force for both the APEX samples and the cryotomography experiments. And the rest of my lab, you see, quite diverse in terms of where we come from and what we do. People at NISB who have helped us along the way, Thermo Fisher, Angela, and others, Nie Xin, for help and guidance and discussions, funding, and thank you for listening. And I hope there are questions later that we can address, and then I can share with you some more sort of technical insights if you wish. Thank you very much.
Okay, thank you very much, Dr. Alexander Ludwig, for your insightful presentations. So before we can discuss with our colleagues here in BRIN, later in the sharing session, discussion sessions, I would like to welcome first our third speaker, and then after that we can have like discussion with all of our colleagues. Thank you very much, Dr. Alexander Ludwig. Okay, we already heard the presentation from our second speaker, Dr. Alexander Ludwig. I hope that all of my colleagues here in BRIN can gain a lot of knowledge, and maybe later, if you would like to discuss with Dr. Alexander Ludwig as well, welcome. We are very welcome, and you can either directly engage in discussion in this Zoom meeting, or you can also write your question in the chat box. You can choose whichever you like. So we already arrived in the third sessions. I would like to welcome our third distinguished speaker, which is Dr. Yudhi Nugraha. So Dr. Yudhi Nugraha is our colleague as well from BRIN, from the Research Center of the Molecular Biology, Ekman. He was graduated from the Nara Institute Science and Technology, NICE in Japan, and also take the postdoctoral in the Spain as well as now in the Italy. And then basically he is a crystallographer as well. So we hope that we can gain a lot of knowledge as well from him. So I would like to welcome Dr. Yudhi Nugraha.
Yeah, thank you, Mbak Fina. So,
Yes.
thank you, Dr. Alexander
Time is
Ludwig,
yours.
for the presentation, and also Nie Xin. It's very clear about the cryotomography, the thing that it's very new for me actually, because I'm a structural biologist based on the protein. But let me share you my screen. So yeah, so here we go. Today, good morning everyone. My name is Yudhi, as Ibu Fina. I'm from Pusat Riset Biologi Molekuler Ekman under BRIN, and also I mark here because Cryo-Em Laboratory is belong to Deputy of Infrastructure and Research Innovation in BRIN. So today, actually, we will provide you very important, I think, about the view of first, so we would like to provide you a brief overview of a Cryo-Em facility in BRIN, and also it is just to shed light what is in the future and grand scheme. It is the aim is to add collaboration in the field in BRIN, of course. So firstly, here in the part one, I will talk about the Cryo-Em facility in BRIN since we start in the last year in 2023. So before we share you what inside our laboratory that consists almost 24 people now, including Pak Rifki here. There is Pak Rifki as head of Cryo-Em facility. And also here we have so many people from Cryo-Em laboratory, including Prof. Dede is a manager for Krios and Tundra now, shifting and also helped by Bu Fina. And also we have Pak Arbi, also present in this meeting. He is working on Aquilos, both with together in line with the Talos. So all of the thing here, the facility is belong to the Deputy Research Infrastructure, as we mentioned before, and it can be utilized by all the research organization in BRIN. It is just brief refreshment that our facility is an open platform that can be utilized not only by researchers from BRIN, but also from Indonesia, and regionally can be used openly with a collaboration scheme. So this is the introduction of a Cryo-Em facility. Now, actually, we have at least 3 microscope of TEM and also 1 SEM Aquilos 2 that just now we talk about. But here I will share you about widely what we have, all the thing. First, we have Talos as a basic TEM, transmission electron microscopy. with voltage to 100 kilovoltage. It can be used not only in room temperature, but the advantage of this talus is a cryo sample. We can freeze it for the sample that very not stable, for example, for life sciences sample. And if you have a sample, if this need in the only room temperature analysis in the under microscope, we can use only directly to talus. For example, you want to see a virus or something that quite big, you just put directly the sample to the fit robot to vitrification. It is to prepare the sample to be in the cryo condition to protect from the electron beam. And then we can directly go to the talus. In case of the single particle analysis, we use using Tundra for primary screening, and also we also can use this Tundra to do determination, even though in this Tundra have limitations compared to the big Cryos G4. So this G4 is very good for a single particle analysis of one particle of protein. And also, we already heard about many good things about what is Aquilos, and I will not add more because I'm not in specific this scope. But I think we have all the things, including the 3D reconstruction using HPC. We also have here Mbak Lina as a manager for this high perform computing dedicated for Cryo-Em. So we know that open platform in BRIN, we can access from ELSA. We have now about 14 services that can be directly opened using the ELSA. And of course, we still add more. There is more additional idea that coming from, for example, Aquilos, Parbi proposed many new services, and also Prof. Dede also provide very good idea about Tundra can be visualized directly. So working on the structure 2D determinations or just imaging with under that one. And also Talos also so many things. And we think that 3D reconstruction using HPC also can be open platform, that someone that have a micrograph from outside the BRIN also can utilize this one in the future. Actually, we can see this surfaces in the ELSA. So, but there is two kind of the surface that now because the utilization of Talos is so huge, it increased day by day. But the utilizations of Cryo-Em facility mainly for single particle analysis and also cryo-electron tomography is still low in our lab. In the future, we also want to develop a microelectron diffraction with the idea to do crystallization as its initial crystallization. And also we diffract with the electron, but it is maybe in the few years in the future. Now our focus is a single particle analysis and also cryo-EM for tomography that already told very clearly by Nie Xin also, and also by Dr. Alexander Ludwig. So also we have we're trying to establish the data storage at Cryo-Em, collaborate with the HPC lab, because thankfully Parifik also working with this high perform computing. So it is working together with Mbak Lina and the team working with this. And in the future, we also trying to escalate the capacity of the Cryospark, and also the GPU what we have. It is dedicated GPU. We'll establish this year for only Cryo-Em. So the concept of Cryo-Em itself, it is just to remind you that it is basically the electron microscopy. As we know that, for example, our sample just freeze it in liquid nitrogen, in ethane, liquid ethane, and then this condition will protect this one, and we put the sample in the electron microscopy, and with the help of the AI working together with the determinations of one single protein, For example, this is single particle analysis, will be analyzed and combined together with a billion or million particle. We call it micrograph. From micrograph to be particle, we extract the particle and we do determinations until we got 3D reconstructions of this. So this is the idea. We just if we obtain the protein and then we put it in the grid and then we see the micrograph and we combine together this to be 2D classification and will be the basis of a 3D reconstruction, and we have the model of protein. Until now, we have almost four very high resolution in single particle analysis, but it is still quite low compared to our target in this year. So we propose we trying to identify the problem of single particle analysis services. So in the future, we can have so many sample for SPA. The second one that today's seminar talking about, it's a cryotomography. I think it's already clear. I think what cryotomography itself, but also it's still low. The sample that come to us to do the cryotomography, especially if we want to identify the architecture of cell, and also if we want to identify the protein located in our cell and localization or something so on. So here actually our cryotomography will combine together the work with the cryos to see the images, not only with the aquilus, but also after lamella will already finish the making lamella, we bring this lamella to cryos to do the taking a picture of this, and we do the construction of the 3D of the cell with Amreta, the name of software. So the Cryo-Em application, I said because I heard some idea that why are we establishing the Cryo-Em facility in BRIN? It is because the major of scope in BRIN, for example, in life sciences, can utilize this Cryo-Em in the future. So it is like a big hope that we can understand the mechanism in the cellular and also molecular, even like atomic resolution. So it is to answer your all research experiment and to understand deeply about the mechanism. So this facility can be utilized, right? So the part two, I would talk about the progress and ongoing and future plan of our laboratory that we start together in 2023 last year. We already installed the instrument with help, of course, from Thermo, and also we already open protein expression lab system for source single particle analysis. But now we are trying with this for purification. And also we also already conducting the training program for the manager and also internal management, because as we know the expertise from BRIN itself and at the beginning of the establishing Cryo-Em facility is very few people working with the Cryo-Em, but we already have now the good team and also the solid team to working together to establish more regenerations of people working with the Cryo-Em. As now we about like establishing the operator for like young generation that in the future we hope they also stick on the Cryo-Em major. So also we already launching the service open it in ELSA. It is just start actually after establishing the installations. We just start on June 2023, and also we have a training for potential user in October. We already do the training, of course, with help with the generosity of Thermo. They send us like a expert from Australia, from Singapore, and from China at the time, the 20 people selected from all university in Indonesia, we joined together with this workshop. And also we already start the global partnership with several university and institute. I can share you the number. And also we already got the structure actually from single particle analysis, even though it's still low number, but we already have initial data for this, and we propose this one writing the paper now. So into 2024 this year, we try to exploring the possibility of determination, and also we try to make a connection between BSL-3 facility because in the beginning of the Cryo-Em establishment is the idea is to make we are ready in a pandemic preparedness. So we will not forget about the COVID-19 era. So we're trying to establish the BSL-3 that we have in BRIN nearby a Cryo-Em facility. So it is just one connection in the future. Also, we also trying to strengthen a collaboration with Asia country and also European structural biology. We are working with Cryo-Em. Now I'm in Italy now, trying to connect the laboratory, our laboratory in BRIN and my laboratory in Italy now. And also we already established the community of Structural Biology Association. We call it ISBA, Indonesia Structural Biology Association. And we also in the future, in the few months, we initiating training program for regional user in ASEAN. Of course, we try to see the opportunity of collaboration. So in the future, we have 5 years plan skills initiative for trying to establish not only for a few years in BRIN, but also in the future in long sustainable years for Cryo-Em facility. As we know, there is idea with ELSA point and also PNBP in Indonesia. We got a start on June until December. It is about like 6 months working with this. We got about 545 million rupiah. But in the future, we trying to establish this month not only to catch up the number higher, but also the higher utilization and also the higher impact factor in the scope of structural biology. So as we know, we have a workshop single particle analysis of protein in last October. So in the future, we will establish more the workshop like this, including the cryotomography. And also we trying to introduce what is structural biology in Indonesia using like a kind of series in webinar. As long as I remember now, we have and this series is number already 9, I think. And also we have also the platform in OR Hayati and Lingkungan. It is like about every week we have a webinar telling us about what is structural biology is. So it is like trying to catch up to invite all the people that work together and that can utilize what is the approach of structural biology can be used for their research. So this is the collaboration in making that we have and start to do the collaboration. And the party, this is I think the most important one, the grant collaboration and scheme that we provide for you all in the future, not in the future, in the few months, I think. So basically the BRIN policy and infrastructure, we know, we understand together. It is like special for Indonesian people. We already know that this infrastructure is working on deputy of infrastructure. So the deputy infrastructure research and innovation working with another deputy to provide the facility. So we will not, for example, DIRI example, we will not give you the fresh money for grant. So it is will be utilized by another deputy, for example, deputy for facilitation. It is have a funding there. So we already communicate and then also under the suggestion and information that I got, in the near future, we will establish the scheme, the program. Usually we call it a platform. And also we have a research organization, a research center, or we call it organization research, OR, and also PR, that can utilize with this. And we also have the basis of research innovation facilitation. For example, we have a postdoctoral degree in BRIN. Also, we have RIM, Riset Indonesia Maju, and also we have research collaboration. But special for this Cryo-Em facility, we trying to make the platform, because the few number of the utilization of the Cryo-Em facility. We're trying to identify what is the problem, why the Cryo-Em facility in BRIN is still low user. So it is because the bottleneck is a protein itself. We have no protein production unit in Indonesia. I think people working with the protein mainly in Indonesia, taking from the natural research and resource, then they trying to purify. In case of this Cryo-Em facility, of course, we need huge amount of protein. Even like compared to X-ray crystallography, the amount is not very high as X-ray crystallography, but it is still need more compared to the originally natural resource protein that got from the compound. For example, we need a DNA recombinant reconstruction, and we have to express it, and after that, we can utilize this structural value. So the bottleneck is a protein production unit. So then this year, we're establishing the Cryo-Em facility just in front of our laboratory. We have Actago now. And also we want to make this one-line platform with the Cryo-Em facility. Even this platform, we call it PPU, also can be used by another people, not only for Cryo-Em. So in the future, our dream is to establishing this protein production unit, and the idea from the sample from the PPU can be directly go to Cryo-Em to do the structural determination. So this is now ongoing because we know that the facility is huge. I think maybe in the mid of year we already have it all. And the idea is we already do the workshop for the Cryo-Em workshop in 2023. And in the future, exactly on June 4 to 7, we are trying to establish workshop for cryotomography. So this is there is the preliminary workshop or training for internal user on May for internal management of a Cryo-Em. And then on June 4 to 7, it will open to all of you that of course there is a selection process, and also the selection also usually helped by Thermo expert. And then we will select and invite you all to our facility to utilize this cryotomography workshop, of course with the Aquilos and also with cryos. And the most important thing, there is a call for a proposal. There is two call proposal from first, the scheme is from BRIN itself. It is a funding facilitation. It will open on 22nd of April. So as you know, you can see under the RIM RISNOV website in this one. And also the second one is a call proposal from Cryo-Em itself lab. It is using ELSA base. So the idea is if you selected to this proposal open a call, you will give ELSA point only dedicated to using Cryo-Em facility. So there is two. First, the funding in this grant, and the second one is ELSA based. So this is the RIM platform structural biology collaboration. It's under ORHL, Hayati and Lingkungan. So it will be open 22nd of April until 6th of May. The head of this platform is Pak Fatoni. Pak Fatoni itself is head of Research Center for Applied Microbe. So he is like a Pusat Riset Mikrobiologi Terapan, the head of PRMT. And the selection administration will start on 7th of May until 16th of May. And also there is substantial selection. So in the future, we have this one. And of course, one of you, I think most of you working with the single particle analysis invited to write the ICP already. And then in the future, we will trying to catch up, and then we will open this one. And then we will do the selection. So the idea is the Cryo-Em facilities not only work for the people working with the single particle analysis, but also with the Aquilos we have Aquilos. We have also another opportunity to see the interaction in between cell and also the histology of cell of their interaction. But mainly working the collaboration or the platform is working with the complex protein in between the protein and also protein with another ligand. So we hope in the 22nd of May we can use this one. Okay. The second one, I think the second platform is either from the laboratory UM lab. I will share you because actually initially Pak Rifki as our head want to do the presentation, but maybe he has another agenda. So Pak Rifki itself head of the research computation in BRIN, but also he worked in HPC and as head of laboratory of Cryo-Em now. So the facility of the Cryo-Em now led by Pak Rifki itself. It is like one line together with the HPC utilization, of course, with of Cryo-Em. So the second platform, we call it ELSA by proposal open call. So this is the facilitations for Cryo-Em. So the motivation and also how the ELSA point work, it's we will first why we see the motivation why we establish one is because they say that the expensive of a Cryo-Em in ELSA, it is like about I forgot like 15 million rupiah for a single particle analysis. Uh, Actually, it is like half of the common price, I think in Singapore, even like in my laboratory in Italy, it is like about 6,000 euro. But of course, we do we know the situation in Indonesia. So because of too expensive, there is no one apply. Even like they using ELSA point, and also technical difficulty because preparing sample and also on is very difficult. So BRIN itself understand and realize that we need to boost this problem solving, and we trying to give the opportunity to make it's like free actually, but with the scheme. So the scheme itself, we will give you ELSA point dedicated only for Cryo-Em. So another bottleneck is also preparations of protein. We can use PPU unit that now are trying to establish just in the front of our laboratory. There is Octago also, and also we trying to buy the column. So the ELSA by proposal, it's like we will give you the ELSA point with the account and ELSA. Of course, the collaboration with the outside Indonesian researcher should be together with the Indonesian researcher. It is to provide transfer knowledge and also boost the regeneration of Indonesian structural biologists in the future. And also we have like regulation to provide this scheme. So later we will give you two schemes from ORHL and also from Cryo-Em lab itself. So for further information, you can ask directly to our head laboratory, Pak Rifki Sadikin, or to us in person. Okay, thank you very much. That's all, maybe from me. Oh, I forgot to share the screen. Sorry. Actually there is the second screen here. So it be like this. So Pak Rifki Sadi can give me the slide. It is the second proposal open call based on the ELSA. Maybe later I can give you this proposal. Thank you.
Thank you very much, Dr. Yudhi. Maybe for the open call, maybe you can share to us later, to our colleague you, I mean. So we can carefully read the open call scheme later. Thank you very much for Dr. Yudhi Nugraha for explaining about the open call platform, either in Cryo-Em laboratory or from the research organization of life science. I hope from now, all of the colleagues you here can prepare the proposal or ICP if it's already open later. It's like a big opportunity. Okay, I would like to say one more time, thank you very much for Dr. Yudhi Nugraha for giving us many information, detailed information, and also enlighten us about the facility, Cryo-Em facility, and also the program as well to us, and we can discuss furthermore with our colleagues here that joining our webinar today. So finally, we arrive in the discussion session, Q&A session between our three distinguished speaker here with our colleagues in BRIN. So we already have several questions from the colleague, our colleague you, dear colleague you here. In the chat box, I can see there is already 3 colleagues you who would like to join the discussion with the distinguished speaker here. So first is from the Dr. Endah Puji Septiani. So the question is like thankful for the speaker, and then she would like to ask how to improve cell addition to Cryo-Em grid. So we charging to make negatively or positively charged grid, and last time we did glow charging and coat the grid using gelatin, but the cell did not attach very well. So I would like to invite Dr. Alexander Ludwig or Dr. Nie Xin to help our colleague here. This is like very technical one, I believe.
Yeah, I think so. I think from this question, it looks like the customer want to culturing the cell together with the EM grid, or maybe they want to apply the samples. Because for the sample apply, for example, like vitrification process, is require quite a bit strategic because the how the filter papers was attached to the TM grid need to be very careful. So, but I have one paper later I can share with you. So they give some suggestions, especially for the side which touching the cell, they will using the Teflons instead of filter paper. They change the filter paper to Teflons. So we will remove any possibility that the cell has been removed by the filter paper itself. So, but in case of the culturing the cell together with TM grid, maybe Alex, you have more experience than I do. How the cell was grow on the surface of TM grid successfully.
Yeah. We have tried different cell types. We've had really no major problems with fibroblasts, with these epithelia cells that I shared with you, others typical like HeLa cells, things like that should actually, they don't really mind so much. They normally grow well on the EM grids. What we typically do is we coat them with fibronectin. I understand what the person tried here was gelatin. We haven't tried that. Normally we coat with fibronectin. For some cells that might actually help a lot. And we have a thin film of carbon, of course, on top, but that is typically, I mean, normally the case. So, and we glow discharge, yes. So the fibronectin, maybe polyl-lysine also might help. Gelatin, I'm not entirely sure, should also help, but maybe polyl-lysine or fibronectin would be the better choice for many of these adherent cells. But it depends a bit on the cell type. Some might not settle so well on the grids.
Thank you very much, Dr. Alexander Ludwig and Dr. Nie Xin. So I would like to invite Dr. Enda. Maybe you would like to discuss more, for example, like what kind of cell you intend to prepare, for example. So give us more picture about your questions. Dr. Enda? Can you hear me, Dr. Enda? I hope you still joining us. I hope yes. I hope the answer for Dr. Ludwig and also Dr. Nie Xin already satisfied Dr. Enda, or maybe we can discuss more later by contacting the speaker directly, either by email, if you would like to discuss more. Thank you very much, Dr. Enda. So I would like to move to our second colleague. You would like to discuss more with the distinguished speakers. So this one is from Dr. Wahyulia. So the question is maybe to Dr. Yudhi, this one. How do I register for the workshop in June? Is there already detailed information about that?
Yeah, we will share the information later. Of course, it's nationally. Usually we collaborate with another deputy that share the information. For example, the last workshop we did sharing nationally twice, and then maybe we can provide you detailed information. Now also we trying to do the internal training on May. So in June, of course, we have the idea with this. Yeah.
Okay, Dr. Yudhi, thank you. So stay tuned, Dr. Wahyulia. We will share more information later. Okay, hope that it helped. So we move to our third speaker with Dr. Syah. So I hope you still in here, Dr. Syah. So the question is for Dr. Nie Xin. So the first question is, can we get SAED image, elemental percentage, and mapping like conventional HRTEM? And the second question is, can we use magnetic nanomaterial plus bacteria or fungi without any problem?
Oh, this is a really nice question. But the first question is whether we can understand the elemental information. So I have to say for Aquilus, we have so far, unfortunately, we do not have a EDEX detector. So which means we do not get in the elemental information directly. So this is a part of the reason is because for the elemental mapping, we need to focus the beam on a particular area for a long time and to induce the X-ray excitations. So in this sense, we will damage the life science materials, which is really soft and under the cryo conditions. So at this moment, we do not have a very widely used elemental mapping capabilities as the Aquilos machines. And for the second one, I have to be very frank with you. So the bacterial fungus is no problem. But for magnetic nanomaterials, I'm not 100% sure, but I do not think it's very likely to be work with Aquilos so far, but I can go back and check with you whether there's any capabilities for magnetic nanomaterials together with cryo meetings. But it's good because I think a lot of people want to understand that whether nanoparticles, like nanoparticle distributions or maybe delivery system with nanoparticles, this is a very hot topic so far. Utilizing nanoparticle as a delivery system for the maybe drugs or maybe inside the host cells to see some of the treatment together maybe with antibodies. So I can refer these questions back to our application teams to help you understand whether the magnetic nanomaterials is available in Aquilos and also the Cryos.
Okay. Thank you very much, Dr. Nie Xin. Dr. Serta Wapsal, would you like to discuss more, or it's already satisfied you, the answer from Dr. Nie Xin? Okay. All set. So thank you very much, Dr. Nie Xin. Then I would like to invite our other colleague to join the discussions with the distinguished speaker here today. So while waiting, actually I have a question related to the sample preparation. So I mean, maybe because like this is like a very new in here in Indonesia, like maybe our colleague here is already have the sample either with this, whether it is cell, virus, or cell culture or for plant maybe, but actually is there any like specific requirement before like we send our sample to the services? I mean, like in the case of SPA, like there are specific requirement, like the size of protein, it must be pure or something like that. So how about in Cryo-Tomo? Is there any such things?
Yes, Dr. Fina. So thank you very much for this next question. So I want to share with you this slides which I presented just now. So in one of the slides, I prepare a small list of samples which are suitable for the Cryo-Em, especially for this Cryo-Em workshop. So can you see the screen I shared over here?
Yes, can.
Okay, good. Yes. So for the samples, actually, tomographies can fit a wide range of samples. So for example, like virus particles, purified virus, some protein complex, even the apoferritins, which is a benchmarking protein, can collect the tomography. But for these workshops, I think the focusing more for the cellular structures, especially designed for the cell biologists. So I will suggest people who want to submit the samples to provide more like a suspended cell culture will be most easier to handle. Because in this case, the suspended cell culture no need to grow together with the TM grid. We can apply the sample directly on the virtual board, which is similar to the single particle data sample preparation. So in this case, let me see. Yeah, so there is a certain criteria for the sample provided. So the first one should be the thickness of samples, because for the culture cells, normally, I think most of cells is like flat, so it's no problem. But in some particular cells, which is really huge in the dimensions, more than 10 micrometer thickness takes a longer time for the film milling to process. So we'll suggest the cell size to be less than 10 micrometer in the thickness. So just to be clear, this is not the dimension of the cell, because for the some of the mammalian cells, the dimension can be maybe 20 to 40 micrometers in the size. But I'm talking about the thickness. Like HeLa cells can be really huge, but in the Z axis, the XY can be large, but the Z axis, the thickness normally is not just big. So most of the cells should be okay. And also, as I said, suspended cell culture is preferred. For example, The most likely sample we're using for demo purpose is like some of the yeast cultures, if it is a suspension cell cultures. The size is in the range of maybe 5 to 10 micrometers. Then it's a perfect samples for this tomography sample prep. But if you particular, for example, as Alex, his examples, he's culturing the cell on the surface of TM grid. So this will require additional steps just to culturing your cell cultures together with the grid. And of course, on the surface of TM grid, you need some particular treatment just to make it more adhesive to the cell cultures. So for this, I would suggest the customers will be, maybe you just send out your proposals and our application specialist can discuss with you how the sample should be prepared, particularly for your particular cell types. But for some of the big samples, which is too sick to be vitrified directly with VitrBot, then you need a high pressure freezing. So in this case, we will not take samples which requires a high pressure freezer because it's more complex for the workflows. So in general, for these workshops, we will suggest people to submit suspension cell cultures or maybe cell growing on the surface of grid. So these will be the easier samples to handle in this workshop. Okay, so Dr. Fina, do I answer your questions?
Yes. Thank you very much, Dr. Nie Xin. I hope everyone here already take notes for their sample to be prepared later. Okay. Maybe I also have some question to Dr. Alexander Ludwig. If like our research is about the interaction between the protein with drug in the cell, like maybe you can give us like how we prepare that kind of sample, Dr. Ludwig, or how we analyze that kind of interaction in the Cryo-Tomo.
Yeah, okay, so a drug targeting a particular target inside the cell, is that, do I understand correctly?
Yes, correct.
Whoa, yeah, that is challenging. What would be the target would be my first question. I mean, maybe you are aware of recent publications where people targeted the ribosome with specific antibiotics and they determined high resolution structures by tomography and subtomogram averaging. That is, to my knowledge, one of the few, if not the only, demonstration that a small molecule target, I mean a small molecule, a drug, was visualized together on its target in a cellular environment. I mean, the key question is, what is the target? And can you identify the target? And can you determine a subtomogram average structure of your target with the molecule bound? And of course, for objects that are abundant, like the ribosome, that can be done. It's challenging, but it can be done. For things that are not so abundant or more difficult to average, that'll be challenging.
Okay. Yeah, because like many our colleagues you here working with drug discovery as well. So like many questions about that, like how to analyze in the Cryo-Tomo.
Yeah, Nie Xin, maybe you want
So I
to
just add on a little bit on this topic because the structure-based drug design, also the drug discoveries is really a big target, big topic. So we have a dedicated team on the pharma sectors, which is expertise on this topic. And for this, my own comments is there's a lot of pharmaceutical companies who is already integrated Cryo workflows into their drug discovery workflows. Like recently, there's a lot of news on the GLP-1 related drugs, which is insulin regulated like weight loss. So all these drug discoveries is the structure determinations and also the molecule screenings and also target identifications can be done through the Cryo-EM structure, getting structure informations. May not be limited to either tomography or single particle. Sometimes it's a combination of multiple methodologies. But I have to say most of the drug discoveries happens at the single particle level at this moment, because for tomographies is more for the verification of drug response in the whole cells, how the structure changes as the target site. So discovery site is more happens at the single particle, which is using the structure information to understand the drug binding pockets, all these small molecules, all these informations. So I think that's the current situations for the drug discoveries.
Okay, thank you very much, Dr. Nie Xin, and also Dr. Alexander Ludwig for giving us enlightenment in that area. So we already have for question from our colleague. It is from Dr. Francisca Mariani. So the question that this cryo-EM is very new to yes, it is very new in Indonesia as well, and like she hasn't purified the protein, not yet now, and then due to what she do right now that work for biological activities from plant material against bacteria or human cell lines, for example, cancer cell, or to apply wound healing in vitro. Is that such insight to do next with the cryo-EM technology, such as to know how the cancer cells itself can be inhibited to grow by the plant sample treated? Will it valuable information that important to do the pricey research have to spend? So maybe all of the speaker here can give the insight to Dr. Francisca Mariani.
Maybe I can say something if I understand. I'm not entirely sure what the goal is, and I think that is always important to consider. What do you actually want to know, and what is the best approach to get to the answer? So anything, I mean, the kind of things that Nie Xin and I, especially when it comes to the cellular cryo-EM approaches shared with you today, they are quite tedious. It sounds always so easy on paper and also straightforward, but in fact, especially when you have very little background in EM or in handling cryo samples, you spend quite a lot of time learning how to handle these things and how to acquire the data and to analyze the data. And if you come with a structural question, then you also have to know how you would want to then finally get to the structure in the end. So we as cell biologists, we are actually already quite happy to have tomograms under cryo conditions that show us things in a native form that we had previously only been able to look at in these plastic embedded stain samples. So for us, that is a major leap. And if there is a question like here, for example, cancer cells or interaction of bacteria or with a target cell or the impact of certain drugs or compounds on a particular structure within a cell, then there is a question that can be done. Beyond that, it's then the question, what is it really that you would like to know? And is the cryo-ET in situ approach the right approach? And how long would it take? And how much investment would that take? So, yeah, even for us, I would say we came with a lot of EM tomography background. It took us a while to get the cryo work to work. It's not trivial, and a lot of things can go wrong, and in fact, they do go wrong. They do go wrong all the time. So what we always present is sort of the tip of the iceberg, the stuff that works, but there is 60 to 60% of the time when we collect tomograms, something is wrong. The sample wasn't frozen properly. There's ice on the lamella, or the lamella breaks. The target is not contained in your lamella, so that is particularly troublesome for correlative work when you're really trying to find one thing in your cell. So there's always things that will go wrong. So the question is, what do you want to achieve? And that's the key thing. What do you actually want to visualize? And then, of course, have the time, invest the time to do that, and the manpower, and
So we need to set the goal first.
Yeah, I think it's important to be realistic about this. If it's a long-term investment for a particular lab or a project, and the manpower is there, and the training capacities are there, and there is a long-term vision, then of course it's important. It's good to invest this time.
Yeah, it is.
Short-term thinking is not a good idea here, I think, because it's not straightforward.
But we still can take a note, Dr. Alexander Ludwig, like we need to set the goal and then form the team, like collaboration as well. And then maybe from there, we can like make planning from there.
Yeah. So Dr. Fina, so just from
Yes.
my insight is for this particular project, which is looking at the plant material against the bacteria and the human cell lines. I think this will be a combination of multiple methodologies. So in a common cell biology labs, a lot of people doing like fluorescence imaging together with confocals or maybe super resolution imaging. Then you can get in how the cellular response to different maybe plant extract or maybe natural product from the plant, how the cell or maybe bacteria responds to that. So once you understand the basic cell biology informations, then you see, okay, I have my control group, I have my wild type, how the cell would respond to different treatment. Then if you want to understand really deep, much deeper insight for the mechanisms, how at the structural level, at the organelle level, how the morphology changes. For example, I have a couple examples we shall discuss maybe in another topics, another webinars is like mitochondria. So under the stress conditions, how the morphology of mitochondria will change. So under the confocal images, you are able to visualize maybe you can do some countings, you can do some measurement of membrane stainings. But under the electron tomographies, you can see much higher resolutions. You can see the higher details to see how the like inside the cyst or changes of the mitochondria under the stress conditions. So probably just echo to what Alex mentioned just now. You should have some a target for imaging before you really jump into the field. You understand that this is the project, this is the particular organelles I want to understand how this responds to particular treatment. Then the tomography can give you much higher resolution and much higher details of your mechanisms behind all these changes. Then there'll be additional information provided for the whole project.
Okay, thank you, Dr. Nie Xin. We can take a note from that. So I hope Dr. Francisca Mariani, is it already answer your questions? Dr. Francisca Mariani. Okay, I hope that it's already answered the question from our colleague you here. We can take a note several recommendations that already explained by Dr. Alexander Ludwig and Dr. Nie Xin. So we can move to the fifth question here from Professor Dede Heri Yulianto. So the question is addressed to Dr. Nie Xin. Could you please explain more details about the high pressure freezing? If we need to extend our capacity to analyze tissue, cell, or bulk tissue, what kind of equipment should be prepared here.
Oh, okay.
Please, Dr. Lee.
Yeah, no problem. So please allow me to share with you the slides. Actually, just now there is a tiny corner of my slides which mention a little bit about the high pressure freezing. So let me zoom in a little bit. So Dr. Fina, can you see the slides over here?
Yeah, clear.
Okay, good. So actually, the high pressure freezing is another method to prepare to vitrify your samples, especially for the large samples like tissues or maybe some organelles, biopsies like from patients, from mouse. The most commonly samples we're using is like the brain tissues from mouse tissues, or maybe the C. elegans, which is a very large multiple cellular organisms. So the limitation for the vitroport is that the thickness of your samples has to be limited. Like I mentioned, maybe less than 10 or 20 micrometers. Because if the cellular tissue is larger than this particular thickness, so they cannot be perfectly vitrified. So in the center of your tissues, they still remain the crystalline ice instead of vitrified ice. So in this case, we need the high pressure freezing. So this is a particular machine that's provided by some other parties. So I think on the market, maybe 2 or 3 suppliers can provide this high pressure freezing. So the most maybe easier one to talk about is from Leica. So they have this. So Alex, is there any high pressure freezing in NTU? I think maybe there is a one in NTU.
No,
you don't
we
have.
don't.
Okay.
We don't have a high pressure freezer at NTU.
Okay, so NUS, I think EMU has a one high pressure freezer.
Yeah.
So in this case, this the machine in size similar to what the Aquilos is really a standalone machine. Your sample will be zoom in a bit further. Your sample will be placed in this particular planchette. We call the HPF planchette. So together with a buffering or maybe some cryoprotectant, will mix with your tissue samples over here. And this sample will be frozen in milliseconds under extremely multiple like atmosphere pressures, extremely high pressure. So in this case, the water will also become vitrified, which means amorphous. So sample will maintain close to native conditions to its natures under the high pressure freezing, because everything was frozen in the large volume in milliseconds scales. So in this case, once your sample is ready, it will be looks like a piece of ice cube because it's not only one layer of cell, but it's also in a large volume. So this sample can be transferred also in Aquilos. So now give me a second. Let me find a suitable presentation for this example. So because in Aquilos just now, what we shared just now is to how we can load one of the TEM grid into the Aquilos. But actually this Aquilos is really versatile, which means they're also able to accommodate even the high pressure freezing samples. Give me a second, let me open this one. Stop sharing. So which means you are able to select different adapters, or we call the cryo shuttles, for Aquilos different samples. Dr. Fina, can you see the slides shared over here?
Yes, crystal clear.
Yes, great. So you can see that you can choose. So normally we are loading the samples with autogrid. So we have this dedicated autogrid shuttle. You can put 2 of your TM grids over here for all the workflows we shared just now. But of course, you can choose the different shuttles. Then you can place different planchette or even the sulfate disk on the different shuttles. But in this case, it's a slightly complex workflows because the volume for the high pressure freezing samples is huge. So you cannot prepare a lamina directly over here. So what you need to do, you can see next to your high pressure freezing planchette is another TEM grid. So you need to you perform the cryo leaf out workflows, which means you cut your samples into a volume. You leaf out a particular area of regions in a large volume, and do the film millings on this we call the bulk laminas. So in this leaf-out workflows, you can transfer samples from your hyperfreezing samples to the TEM grid, we call the half-moon grid. So you can see one half of the shuttles will be punctured, the other half is the TM grid. You pick up one volume of the high pressure freezing samples, transfer to the TM grid, and you perform your cryo-feed millings over here. You also can prepare laminas. You can collect your tomogram on this polished cryo lamina as well. So this is a slightly complex workflows because there's one additional steps needed, which is a cryo default. But utilize these workflows, you can access a much larger volume. For example, some larger samples which cannot be frozen directly on the TM grid, but need high pressure freezing machines. You can image in some particular areas within the tissues, maybe not on the surface of the tissue, but maybe something deep inside, even like the some area which we cannot access directly. You can using this cryo default to access these particular areas. Then do the feed millings, then you can produce the lambda and do the tomographies. So this is requires a dedicated shuttles, which is can accommodate the high pressure freezing shuttles. And you need the standalone high pressure freezer machines to the sample preparations. And you need to perform the cryo default. But combining all this together, they can produce also the cryo laminas for the tomography data collections. Okay, so I think this is basic information for the high pressure freezing samples. I hope they give you some basic understandings. So this is the additional slides I can share with you is how we can combining high pressure freezing together with fluorescence informations. Because high pressure freezing is a kind of much trickier. What you see is just a huge volume, but you cannot see too much details of your samples. So in this case, if you combine the integrated fluorescence light microscope, because the iFilm, then you can, based on the fluorescence information, you can locate, okay, this is the particular region of interest. I can do the cryo default. So I can cut up maybe 1, 2, 3 different areas, do the cryo default, and produce the cryo lambda as these 3 particular area, which contains the fluorescence information. So make sure that when we do the default, we are not doing blindly, but we are doing particular area based on the fluorescence information. So you also can verify your fluorescence information while you're doing the feed millings. Make sure you always can preserve your fluorescence information within the laminar areas. Then you can make sure your tomography is imaging the right area of your interest. Okay, Dr. Fina, so this is a very general information about high pressure freezing samples, how we can handle that.
Okay, thank you very much, Dr. Nie Xin. I hope it helped, Prof. Dede.
Yeah, thank you very much, Dr. Nie Xin. I think this is very good information for us, and I hope that we can accommodate the research on tissue cell. Actually, I think we need to discuss more detail with you and Dr. Yudhi and also Pak Albi maybe about how to facilitate this research. I think. Yeah.
Okay.
Dr. Nie Xin.
Okay, thank you very much, Prof. Dede. So actually, we still have time for discussion. So while waiting from our colleagues to join more on the discussions, actually, I have one questions like I would say that this is like a mature question because like many of us here is pretty much clueless. What can we expect? I mean, what kind of data that we can expect if the data that we got from the Cryo-Tomo is categorized as already a good data? Is there any parameter that we can say, yeah, we already get the expected result from the Cryo-Tomo? Is there something like that, Dr. Nie Xin or Dr. Ludwig?
Yeah, I think that's a good question. So I had actually prepared a few slides. If I can share my screen again, hopefully this time around without major hiccups. Where is it? Yeah, here. You see that?
Yes.
So, Nie Xin, please, add to it if you have anything else to say. So, I think there's some things that are worth mentioning. I already alluded to it, that oftentimes when we give these talks, we of course show what can be done, but what is actually happening on a daily basis is often sort of a bit ignored, and there are things that can go wrong. So I had sort of, for the presentation, thought about these things that can go wrong and have been going wrong for us, for example, in the past. So I already mentioned that these lamella, they are quite sensitive, so they can break. Sometimes they have for example, they are not completely gone or broken, but they're sort of split in part. In fact, funnily enough, you can still image from these lamella, even though they are not intact, as long as they are ice-free and they look otherwise okay and relatively clean, they can actually still be used for data collection. Ice contamination is a general problem. Should it be too much, then one should probably move on and not image. It's a bit of a waste of time. It's hard to avoid altogether, and I suggest to avoid long storage times in between imaging and move from data sample preparation, so the Aquilos part, to the tomography part as quickly as possible. Of course, always having in mind that the data handling during the transfer of the sample from one instrument to the other is really important, and the freezing conditions for each sample need to be optimized. So what works for one cell type might not work well for others. And so the ice thickness, of course, needs to be good. It shouldn't be too thick for plunge frozen samples, and it shouldn't be too dry either. So for each sample and cell density, there is going to be some degree of troubleshooting what works best for that sample. I can also suggest that, especially in the beginning, don't prepare too many lamella per EM grid, rather go for quality than quantity. So to get more familiar with the milling procedure and maintain your lamella that you have prepared in a good condition, rather than aiming for many lamella and then have the sample in the Aquilos for maybe too long, and then other things can happen while it's in there. It's stable relatively, but of course, the longer time these lamella are exposed to these conditions, the more things can happen that might not be so good. I will share with you some things that might not be so easy to spot or in the lamella. One thing is evidence for ice formation inside the sample, or freeze dry. So what do I mean by that? So here, for example, you see a comparison of what I think is a fairly well-preserved sample on the left, and a freeze-dried sample on the right. So a freeze-dried sample, you will be able to tell something is not right by looking at the membrane architecture or membrane structure of your objects. For example, here you see a mitochondrium, and you can tell that these membranes are not well preserved. You have edges, you have partial fusion of the outer and the inner membrane of the mitochondria. That is not normal. Here on the left-hand side, however, you can see that this is ternary, are well preserved. The membranes are smooth. The other thing you can observe here in the image on the right is, for example, up here in the top right corner, magnified down here, is what is probably ice improper freezing of your sample. So you see here these striations. That is difficult to spot sometimes in the sample before you image, and also sometimes in the tomogram. It requires a bit of an eye developing an eye for these things. So these are this on the right here is not good. A similar example is here in the nucleus. So on the left is a good example of one of these tomograms that we've recorded. You can see here the nuclear envelope, the nuclear pore, and then here the chromatin, patches of chromatin associated with the nuclear membrane, the envelope, and here are zoom-ins. And here is an example of very much a similar region on a different lamella, perhaps even from the same grid, but a different lamella, and you can tell that something is not right here. And this is typically, or can be, the result of freeze dry. That means something made that area on the grid warm up slightly and then freeze again or not. So it's partially degraded. You can tell again, the nuclear envelope is not straight. It has these edges. It looks very rough, not healthy. And the chromatin is also sort of, yeah, it looks funny. I can't even describe it. It doesn't look good. So these are the kind of artifacts you can obtain. There may be others, but it's good to be critical and about your own data, as much as as painful as it is sometimes to discard such tomograms when they're not great. They are in fact not usable. This is not a good sample. Nie Xin, you're more than welcome to comment on this if you like.
Yeah, thanks, Alex. I think this is really useful, especially you show a lot of real examples from your own research. So this is a first-hand experience, like how to see what is good, what is bad. So I want to comment from another angle, actually. This is not really from the cellular sample point of view, but it's from the data quality. Is once you collect your tomograms. So because actually BRIN has this software called the Tomo Live, which will do the reconstruction on the fly. So after reconstruction, the Tomo Live will help you evaluate the quality of your reconstructive volume, including the CPF, all the drift, and all the shift of your images during the reconstructions. So this gives you the first-hand evaluation of your data qualities. It's not telling you whether you are imaging the right positions or maybe your cellular tissue was preserved within your tomogram. But this will give you the first-hand information is the overall tomogram that you collected, from image point of view, whether it's a good quality tomogram or not. So this information you can obtain directly from the TomoLab softwares. They give you a different level of evaluations to tell you whether from the image quality point of view, whether your tomogram is a good tomogram or not. So it's not the cellular information point, but it's more for the images. So Alex, would you mind just let me share with you the images from the screenshot from the Tomo Live?
Yeah.
Okay, let me do this. Okay. So this is how it looks like. You can see this is a Tomo Live. Then they will, once your tomogram data collection will finish, they will do the data analysis and reconstructions directly on the fly. So you are able to see, okay, this is how the profile of each of my tomograms, including the CTF evaluations, all the alignment features, which means between each steps of your tomo series, how much shift they are happening, and also the motion correction values, like how much motion correction, how much drift was happens during the data collections, and how many motion corrections was applied. So in this case, also over here is some of the like defocus values and also the sample thickness evaluations for that. So to give you the first-hand information, including all these CTFs, astigmatism, and all these different defocus values, sample thickness, and also you can put your own ratings to see, okay, this is looks good. I want to further analysis on this, or maybe from the initial part, the data quality is not very good, so I can skip these samples and I can move on to my next samples. Then also they could use some estimations after reconstruction of your sample thickness. You can select the most suitable thickness of your samples before you really look into the contents of your tomograms. So some other information also available. So from this, they give you really a quick screening to see whether my image quality is good or not before I really look into the details. Make sure that whatever you're looking at is at the high qualities. So they give you really a first-hand screening to make sure your image quality is good. I think this is also can be useful for the users to evaluate their tomogram qualities. Because the image quality is very important for the following like subtomal origins. If your image quality is not very good, then your subtomal origin cannot reach really high resolutions at this point of view. But why this is important is because the throughput rate is higher and higher. They used to be spend really a long time to collect 1 series. But now with the new software, the throughput rate, the speed is getting faster. And also the production of lamina is getting much faster. So the limitation is not really how many lamina I can produce today or how many tomograms I can get. Is the limitation is how many high qualities tomograms or how much machine time I can accommodate for this. So now the tomogram quality, image quality has become one critical point in the tomogram data analysis.
Okay, thank you, Dr. Nie Xin and Dr. Ludwig. Like from both of you explained before, like I can take a note that like we have like 2 point of view, like from the quality of sample and also the quality of image itself. That's the 2 parameters that we can take a look. Thank you very much. So I do think that we are very happy to discuss to all of the speaker here, distinguished speaker here, and I believe that everyone here, our colleague is very satisfied with all of the sharing sessions and then the insight that you give to us today. Hopefully we can like take the inspiration from that for our next research. And like before I close this sharing session, I would like to move to Dr. Yudhi one more time about the workshop. So is there any like quota, limited participant for the workshop next June so that everyone can prepare if want to join the workshop on the June? Dr. Yudhi, please.
Yeah, of course, because our laboratory capacity is limited, and also the room also very small. I think it is still under discussion by Prof. Dede and Pak Arbi. I think in the near days, I think we will decide. It is like approximately like 20 people. I don't know how much. I think it's like suitable to our laboratory room. So it is not only limited to the expert, but also the room of our facility. So it is need to be decided. But the thing is, not only workshop, but also for open call, we have about 25, I think. The proposal can be approved for that. So hope everyone can try to apply in future.
Thank you very much, Dr. Yudhi, for the detailed information. I hope everyone already take a notes about the workshop and the upcoming call for proposals. And then lastly, I would like to say thank you very much to all of the speaker here, to Dr. Nie Xin, Dr. Alexander Ludwig, and Dr. Yudhi Nugraha for coming today and share all of the knowledge related to the Cryo-Tomo and give us inspirations. I would like also say thank you very much for the colleagues in BRIN for joining today's webinar. I hope you already gain a lot of knowledge and insight for our distinguished speaker. So I would like to close the sharing sessions, discussion session, and thank you very much. I will give the time back to the MC, Pak Tatang. Thank you.
Thank you, Fina, for leading a very interactive discussion. And thank you to all speakers for a very interesting presentation. Ladies and gentlemen, this is the end of our agenda for today. I am Tatang. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank
Waalaikumsalam
you, Dr.
warahmatullahi
Nie Xin, Dr. Ludwig, and also Dr. Yudhi for sharing.
Thank
Thank
you
you,
for the
thank
invitation.
you.
Thank you.
Thank you, everyone. Thank you.
So see you next time.
Thank you.
Bye-bye.
See you
Bye-bye.
later. Bye-bye.
Yeah, thank you, Dr. Alexander and Dr. Nie Xin also. I'm Francisca Mariani. Thank
Yeah.
you for the detailed information before. Yes.
Okay. Thank you, Francis. Yeah.